It's really a pleasure to be here as part of this meeting and to see all the different stakeholders who are joining us, because I think this is a conversation that we need to have about what are the best ways to move forward with precision medicine for pediatric brain tumors and for cancer in general. Great. So I have no financial relationships to disclose, and I will discuss off-label and investigational use of drugs. That's kind of the whole point of what we're talking about. Pediatric cancer is complex. Pediatric brain tumors include many, many different diseases, and this is just one depiction of some of those forms of the disease. And even within each pediatric brain tumor type, there are many different subtypes. Is there a laser on this? Maybe not. Nope. I just killed it. There we go. All right. So, for example, genomic studies first identified 4 different forms of medulloblastoma, and now there are different subtypes of each of those, somewhere between 12 and 14 different forms of the disease, and probably more to come. Ependymoma, 9 different forms, and again, more to come. And so I think with this kind of complexity, you've got to imagine that there's going to be heterogeneity in response to therapies. And yet, most patients who are diagnosed with a brain tumor receive very similar therapies. They, of course, get surgery in cases where the tumor can be removed. They're treated with radiation, often craniospinal radiation, nowadays, hopefully proton in many cases. And then they're given a cocktail of chemotherapies that have been around for 30 or 40 years. And at the end of this, some patients do survive, and those patients often end up with very severe long-term side effects. And then many patients recur and end up dying of their disease. And so the question is, how do we improve outcomes? So this is just some examples of the poor outcomes that we see: brainstem glioma, or DMG as it's now called. 5-year survival of about 2%. That hasn't changed dramatically, although there are valiant efforts to do so. In medulloblastoma, the 5-year survival looks better, but 10-year survival is not great. And patients who present with metastasis or patients who undergo relapse have very, very poor outcomes. And for all of these patients, the therapy has devastating side effects: cognitive deficits, endocrine disorders, secondary cancers that arise from the treatment. We're really treating with a sledgehammer something that we need to be treating with a laser. And so the question is, how do we get to these more effective and less toxic forms of therapy? And the hope is that precision medicine can help, that we can look at a patient's tumor and figure out what mutations are driving it, and then give the specific drugs that are going to target those mutations and not cause massive side effects throughout the body of these patients. But there are problems with this approach. Many pediatric brain tumors have no mutations at all, or at least nothing that we recognize as an actionable driver mutation. So for example, in group 3 and group 4 medulloblastoma, there's this barren landscape of tumors that have no mutations that we recognize or know what to do about. Ependymoma is another example where there are very few mutations per tumor, and most of them, even if we understand what they do, we don't have drugs against them. And so how do we deal with this problem? The trials that have been done on using precision medicine for pediatric cancer and pediatric brain tumors, often when papers are published, they try to present this in the best possible light. But the truth is, if you look at how many patients have mutations that are characterized as category 1 or as gold or platinum or whatever the scheme is to describe actionable mutations, very few patients have mutations for which we know there's a drug that is going to work. Lots of patients, pediatric patients, have p53 mutations. We don't have a drug for the absence of p53. And so if you ask, is it important to sequence these tumors? Absolutely, because if there is one patient who has an actual mutation and we give them a drug against that and we cure them, that's fantastic. But we need other approaches. We need supplementary strategies. we need to look beyond the genome. And so over the last few years at Rady Children's Hospital, where I was until a month ago, we developed a multimodal approach where we took advantage of multiple approaches to try to look at the patient's tumor. DNA methylation, which you've already heard about, is a really good way to categorize tumors and figure out what subgroup and subtype they are. Whole genome DNA sequencing, which can discover those somatic mutations and copy number changes, and most recently, extrachromosomal DNA. RNA sequencing, which can look at both fusions that sometimes are targetable and at pathways that are activated. And then we've incorporated high-throughput drug screening, which you heard a little bit about from Mark a minute ago. And it's really this combination that I think gives us at least a little bit more power to be able to pick therapies that might be useful for the patient. And then we bring all of these data, if the data are clinical grade data, and if we get them in time, we can bring them to a molecular tumor board where we can talk about what are we going to do for this patient. And so I just want to walk through some of the data types and the sources of these data, because I think it's important to figure out how we're going to get all of these data for a particular patient. At Rady Children's, there's a rapid germline whole genome sequencing facility. They, I think, still hold the Guinness record for the fastest whole genome. I think it's somewhere around 17 or 18 hours now. But so they can do in-house germline whole genome sequencing. And that's very useful, particularly for patients who have a germline mutation that is driving or contributing to their cancer. We've collaborated with Ken Aldapi at NCI to get DNA methylation analysis. This is on the Illumina platform, and then interpreted using the methylation classifier that was developed at the DKFZ in Heidelberg. And you'll note for each of these that they're CLIA certified or CAP and CLIA certified. Whole exome DNA and RNA sequencing, we've done this in a variety of ways. Everybody does this in a different way, but we've collaborated with a company called, well, originally Ashion. Ashion was bought by Exact Sciences, and they have a platform called OncoMap Extra, which does whole exome sequencing as well as whole transcriptome sequencing. The important thing here is that they provide the data within 14 days, and they provide a report that you can look at, and if there's something actionable, it's right at the top of the report. So you can say, this patient has a TRAC fusion, we should give them the TRAC drug. So we get RNA and DNA sequencing, and then we get high throughput drug screening. We used to do this in-house. We can do it very quickly in-house, but we didn't have a CLIA certified lab. At the University of Washington, there's something called the QUELLOS High Throughput Drug Screening Core, and we piloted with them an assay where we picked 175 drugs, about 2/3 of which are FDA approved. The others are experimental drugs. We have a panel of these drugs, and we send them the cells from the tumor, and within 7 to 10 days, they send back a report of which drugs were most effective on this tumor. And I'll show you examples of all of this in a minute. So we've been doing this for a few years at Rady Children's. This is just a list of the cases we've done using this platform. You can see that a variety of diagnoses, including DIPG. So Rady has a robotic biopsy setup. They can get multiple very small cores from DIPG patients, and in those cores are enough tissue to do a drug screen. And so we've been able to do that. DIPGs, high-grade gliomas, choroid plexus carcinomas, medulloblastomas, and so on. And for each of these, in many cases, we've been able to get the methylation drug screening, flow cytometry, and high-throughput drug screening. So if you plan this carefully and if you work with the surgeon and the pathologist, you can get enough tissue to do all of these analyses, and that's really important. So having done this for a couple of years at Rady, I approached PNOC and said, can we do this on a larger scale? And with a lot of work and a lot of discussion, we came up with a national version of this trial. go a little faster then. And it's described here. Josh Rubin is the PI. A number of us are co-chairs. A pilot trial testing the clinical benefit of using real-time drug screening to determine an individualized treatment plan in children and young adults with relapsed medulloblastoma. So we focused on one disease for now, but there's nothing about this trial that is really specific to medulloblastoma or even to pediatric brain tumors. And the real goal is to determine the feasibility of using this approach to come up with a treatment for patients and to see if this is safe and non-toxic. So the trial launched in February. It's currently open at a few sites and approvals pending at others. And I'll just show you an example of how this looks for one patient. So this is a patient with recurrent group 4 medulloblastoma. I won't go through all the clinical details, but you can see them here. This patient received proton radiation and combination chemotherapy, relapsed, and went to surgery to remove a lesion in April 22, which is when we got the tissue to be able to do the things that we've done. And so this is a picture of the MRI. I won't belabor this for those of you who know how to look at MRIs, but this is a recurrent tumor for which there is really no standard. The original gene panel revealed no mutations, nothing actionable, or really nothing at all. And we redid this with Exact Sciences, and again, nothing in there that could be targeted. So we were left with high-throughput drug screening as the only approach to be able to identify a potential therapy. And this just shows the metrics that we're looking at for feasibility. So the sample was dissociated at the site of surgery into a single cell suspension that was FedExed overnight to the QUELLOS Center in Seattle, plated into 384-well plates, drugs added, plates incubated for basically 3 days, and then developed with a cell titer glow assay, which looks at cell viability. And we got the report 6 days after surgery. So we usually get the drug screening data before we get any of the genomic data. This is an example of what the data look like. These are the compounds, the IC50. So the lower the IC50, the more potent the compound. Most of these you can see are FDA approved. There are a couple of investigational. And this is just a subset of the 175 drugs we looked at. These are the ones that have an IC50 below 1 micromolar, which means they're extremely potent. You can see the different classes of drugs, and I'll go through this more in a second. And some attempts to predict blood-brain barrier permeability, which is, of course, really important. Thank you so much. And so what you can see is there are a bunch of drugs that are really potent that kill. If this number is 0, that means it killed all the cells. You can see we're killing somewhere around 90% of the cells with most of these drugs. So these are candidates that we could use potentially for therapy. This is the next most potent set of drugs. I won't go through them in detail, but you can see some of the subclasses: histone deacetylase inhibitors, proteasome inhibitors, anthracyclines, kinase inhibitors, and others. So there are a number of classes of drugs that you could potentially use for treatment of this patient. So how do you pick? Well, we pick based on a number of factors, including potency, permeability, that is ability to cross the blood-brain barrier, availability. There are lots of drugs that are harder to get than others. Drug class, we don't want to treat with 4 different HDAC inhibitors. We want to treat with drugs that have different mechanisms of action, and yet drugs that have non-overlapping toxicities. And so these were the drugs that we picked for treatment of this patient: an HDAC inhibitor, a proteasome inhibitor, an anthracycline, and aromasin, which is an aromatase inhibitor. This is a drug that's not really ever been used for treatment of medulloblastoma before, but we thought the activity looked really good. And all I can say is right now, the patient, we were able to demonstrate that the patient could be treated safely. This is feasible, and this is an approach that we have to test to see whether it helps us predict what drugs are going to be most effective for the patient. So in summary, recurrent medulloblastoma patients often have very few mutations. This is true for many pediatric brain tumors. It's feasible to collect live cells at the time of therapy and process them for drug screening. The drug screening data are obtained quickly and can be used to guide therapy. And so far, this seems to be a safe and feasible approach. So These are some of the people who have been involved at Rady Children's. Real major shout out to John Crawford, neuro-oncologist who really facilitated this. John has left now. Megan Paul has taken over. Ken Aldapi and Lucas Chavez for helping with the interpretation of the methylomics data and so on. And then on the PNOC level, Josh Rubin, the other co-investigators, phenomenal help from both our pharmacologist, pharmacist, and statistician, and the support of these foundations. And so with that, happy to take questions. Thanks so much.